Хорошо, спасибо. Guys, start coming for lecture. Guys, oh, very good. Praveen, you are the first one from the first division to listen, and now I know that the link is gotten, and you may use this lecture in your special microbiology studies. Because, my dears, I would like to remind you that now you are listening for the lectures in special microbiology. And in special microbiology, it is the third lecture. I would like to remind you that the first one was fungi, pathogenic fungi. The second one was Staphylococcus and Streptococcus, my dears. If the lecture Staphylococcus and Streptococcus was not recorded properly, check it. Then on Monday, I will give another version of lecture on gram-positive cocci, staphylococci and streptococci. Because when I read it on the, I think, 13th of May, it was recorded not properly because of the problems with the internet. Okay? So then for better version, so on Monday it will be given in the full version. Okay? This Monday. Then I will leave links everywhere on the department side. Okay? So now, the third lecture in special microbiology is about Enterobacteria. So from the family Enterobacteriaceae. And the only bacteria you know by now is E. coli, and this E. coli is from this family. Okay? So, and you can see the large or with the high magnification photographs of these gram-negative rods. Of course, it is not staining, gram staining. This is false uh, uh, electron microscopy, right? Scanning electron microscopy. So that's why don't look at the color. It doesn't matter. But just the morphological features. Quite often they are seen as cocobacilli or thick fat rods, right? That are scattered. So here you can see their morphology very well. And on the slide here, you can see that they have a lot of flagella, my dears, right? See, they appear retricious mainly. Okay, we go ahead with Enterobacteriaceae. So this is a very large family of enteric. It means those that mainly reside in the human or animal gut, gram-negative rods. They mainly all live, so the natural habitat is the intestinal tract. My dears, not feces like you answering in sanitary microbiology. So, no, they don't like to live in feces if it is outside the human body. They just live in the most probably high or the upper, but most probably lower intestinal tract in the colon. Okay? So that this family really is very large. And it consists of more than 30 genera and about 120 species belong to this genera. So they are really this is really very populated family. So what genera of the family Enterobacteriaceae are associated with the human diseases? That's a very difficult question because quite a lot of them may live in our body as normal flora. And if you take a look at the names, so Citrobacter, Morganella, Escherichia, Proteus, Rasa, Klebsiella, there's a normal flora. But we have to confess that under some conditions, at least some of them, Proteus, Klebsiella, Enterobacter, Escherichia, even Morganella, under some conditions, if the body is immunocompromised or under the long-term antibiotic treatment, these normal flora members may cause diseases. What does it mean? It means that they may act as opportunistic pathogens. Okay? So when the body is immunocompromised, they get opportunity to overgrow and to cause diseases. And but generally in this family, we may have also the bacteria that are considered to be primary pathogens. What does it mean? It means that with no referring to the immunity, whether it is high or low or immunocompromised or very well. So these three, Salmonella, Shigella, and Yersinia, genera are considered to be highly pathogenic, capable of causing uh, diseases of different degree of severity. Of course, Yersinia, especially pestis, is the have highest pathogenicity, but then Salmonella and Shigella are also highly pathogenic. So then, the members of this family may cause the infections located almost everywhere in the body. So, and if you take a look at this picture, if you take a Escherichia, it may cause infections, for example, brain abscesses, if it enters the bloodstream. It may cause bacteremia when they travel through the bloodstream. So then the pulmonary infections can be caused by Klebsiella, Escherichia, sometimes Enterobacter. Then the bloodstream infections, Escherichia, Klebsiella, Enterobacter. The pioneers over the uh, gut infections are Salmonella, Shigella, Escherichia, and Yersinia. So also urinary tract infections can be a result of Escherichia, Proteus, Klebsiella, Morganella infections. Okay? Almost any system can be infected by them. So now we try now to describe Enterobacteriaceae like we usually describe bacteria. Some most common morphological, structural, and cultural characteristics. So, as we stated, they are gram-negative rods, quite small, usually up to 3, not for 6, it is unusual. So, and they are located irregularly, or the arrangement is scattered or irregularly. And these are small gram-negative rods. Some of the rods may have capsule, but of course not all of them. The member that is well known for the capsule is Klebsiella pneumonia. And you can see Klebsiella pneumonia stained by negative staining, not gram-negative, but by negative staining, where the background is stained. Then we also try to stain a little bit, at least the cytoplasm of bacterium, but the capsule can be poorly permeable, so it is not stained really, it stays white. So that's why we call it negative staining, where the background is stained and the object is not stained. So, but generally the capsule, the thick, heavy capsule, is not really very common. And most probably, Enterobacter, Escherichia, and some others may have microcapsule. So that consists of some capsule antigens, but maybe it is not that thick and heavy like Klebsiella pneumonia. And one more very important common features for all Enterobacteriaceae that they cannot form spores. So how about motility? Mainly coliforms are peritrichous. See, the flagella are distributed eventually over the bacterial body, right? And they usually swim. How they move? They usually swim. But then some of Enterobacteriaceae are non-motile, like Shigella. By the way, so Shigella may have a very closely related strains of Escherichia coli, and they even cause the diseases that are similar in pathogenesis. Then the Escherichia that cause Shigella like this, Shigellosis like disease, it is also non-motile. So the structure of the cell wall and the antigen that are found in the cell wall of gram-negative bacteria. You can see here, while this gram-negative bacteria, so there's the cellular inner membrane, or, right, there's a plasma membrane. Then we have a periplasmic space. This is a membrane, and this is periplasmic space. And in the periplasmic space, no, my dears, this is a periplasmic space. Okay. This. So usually we have a periplasmic space where we have a layer of the peptidoglycan, right? That is, my dear, this is not, this is a periplasmic space. And we have here the peptidoglycan, right? See. This is one plasma membrane, then another membrane, and that is the periplasmic space. And then outside the inner membrane and peptidoglycan, and the top of the second outer membrane, that is here, right? We can see a structure that is called lipopolysaccharide, my dears. Do you remember that it consists of three basic groups? The upper part of the LPS is oosomatic antigen. That is the major antigen of gram-negative bacteria. And these are carbohydrate side chains of the backbone polysaccharide core that are usually expressed on the surface of the outer membrane, and they are quite immunogenic, so antibodies can be produced against this portion or polysaccharide or so-called or somatic antigen. Then polysaccharide core, and then lipid A, that is the most toxic portion of this structure. And do you remember the structure is in another way is called endotoxin, my dears. All gram-negative Enterobacteriaceae has lipopolysaccharide with the lipid A, and all toxic properties of the gram-negative cell wall structure are attributed to the lipid A. Okay, so then here you can see that we may have flagella, and they are distributed eventually there, right? And they are usually fixed in the inner membrane. And also we may have pili. Do you remember The very fragile protein hair-like structures, but much thinner and shorter than flagella, and they are not, of course, for movement. So, what are the basic biochemical and cultural properties of these bacteria? So first, it is classified as facultative anaerobes or aerobes regarding the oxygen requirements. Mainly, almost all of them are facultative anaerobic bacteria. What does it mean? It means that they are aerobic. They like to grow in the presence of the air. But if conditions are anaerobic, they still may get energy from the sugars, right? So they still can get much lesser energy than in aerobic conditions, but still they may grow. Uh, how do we culture them in the laboratory? They are mm, nutritional requirements are not very fastidious, right? So quite often they may grow on the simple or basal media as peptone or meat extract media like MPA. That one you know, meat, peptone, and carb, like, right? So and usually they form circular, convex, smooth colonies with distinct ages, but they are most probably indistinguishable from many other bacteria. So usually they grow rapidly, and for their growth we may use non-selective and selective media. When we use non-selective media like blood agar, for example, if we uh, would like to recover bacteria from the blood, we suspect bacteremia. Then the only microbe that is found in the blood, or any microbe is found in the blood, that will be found in the blood, is considered to be the causative agent of the disease. So we don't need to select any microbes there because the blood is normally sterile. If you find microbe there, so this is very bad and this is a causative agent of the disease. So that's why in that case we will use non-selective media, usually quite often blood agar or some other media. But then very often, because quite a lot of Enterobacteriaceae may cause enteric infections in the GIT, then if we take feces as a sample, as a specimen for examination, of course there's a plenty of different other bacteria, right? So then, and we will need to selectively grow the particular gram-negative bacteria that we suspect that when we suspect they cause disease, right? Then we use selective media that prevent the growth of other, let's say, gram-positive bacteria and let gram-negative bacteria grow. And here you can see the example of selective McConkey's medium that is described everywhere in your lecture books, in your protocols, everywhere, okay? So here we have the uh, inhibiting component that is usually taurocolate or bile salts that Enterobacteriaceae can stand them, or some of them like Salmonella even like them. But gram-positive bacteria cannot stand it, so they won't grow here. And also we have their indicator and lactose. So because in the same time this medium is not just selective but also differential. We would like to differentiate bacteria, gram-negative bacteria that can ferment lactose, and on this medium they form bright red colonies. And another one is gram-negative bacteria that cannot ferment lactose. They form the uh, non-pigmented colonies on this medium because they don't ferment lactose. So in this McConkey's medium, in the same time, we get rid of gram-positive bacteria that inhibited there, and also differentiate gram-negative bacteria that grow here into lactose fermenters and non-lactose fermenters. So some other biochemical properties are they are catalase positive. It means they can split carbon, or sorry, they can split H2O2, and oxidase negative, that is distinguishing from other gram-negative bacilli. And they also may reduce nitrate to nitrite. The activities of various enzymes, including carbohydrate fermentation enzymes, are used in biochemical differentiation. And we know that we usually distinguish first lactose fermenters and non-lactose fermenters, and here again, so lactose fermenters look bright with metallic sheen, and non-lactose fermenters appear just slightly pink, right? And then some of them may also produce hemolysis. So, biochemical characteristics are very important to Enterobacteria because the identification and classification of these bacteria are performed based on the biochemical properties that we mentioned and also antigenic properties that we have discussed before. So what biochemical properties are important? Usually, we have 4 tests that initially are used for rough identification of Enterobacteriaceae into several groups, okay? So, this is called IMVIC test. So, I stands for indole production from tryptophan. We have studied this reaction in general microbiology. Then M stands for methyl red reaction. And this is showing the degree of glucose fermentation and the degree of the medium acidification that may occur within 4 to 5 days. V stands for Voges-Proskauer reaction, the production of particular substrate, that is acetyl methylcarabinol, from dextrose. And not all Enterobacteriaceae may do this. So we may have positive, negative also. And the last one is C, citrate utilization test. That's the ability of microbe to use citrate as a sole source of carbon. Okay, you can see the example. So E. coli, first 2 positive, Voges-Proskauer citrate negative. See, that is the formula according to IMVIC test. Klebsiella pneumonia, vice versa, upside down. First 2 negative, then 2 positive. Salmonella typhi, negative, positive, and we may have some other groups. So according to IMVIC test, the Enterobacteriaceae also can be divided into groups for initial identification. Then some more biochemical properties. Ability to produce hydrogen sulfide when inoculated in peptone medium. And that is produced mainly by a lot of Salmonella species. And usually Escherichia and Shigella are H2S negative. Then production of urease enzymes, gelatin liquefaction, some amino acid decarboxylation, and arginine dehydrolyzation, and a lot of others, my dears. When we would like to identify Enterobacteriaceae, and altogether we have around 125 species. That's not so easy. So that's why you can see how many tests, at least 20 are here, we need to perform to do full identification of the microbe. And here you can see the special uh, identification kits that in microvariant. So we put drop of broth culture into each micro volume, micro well, right? And then after 2 hours, we can see the changing of the color, and according to the manual, we may read the result of this test. That's very easy to use, and that's very this much easier to perform identification in microvariant using these specially produced test kits. So, uh, as we mentioned that we use selective media most probably for pure culture isolation and presumptive diagnosis. So we can see endoagar that you study right now also. Because the endoagar is used in sanitary microbiology membrane filtration test. So to rise the colonies of coliform bacteria that can ferment lactose. So this is E and B agar. So because it has eosine and methylene blue, eosine and methylene blue agar. So, and this is selective medium that inhibits gram-positive bacteria, let gram-negative grow. So E. coli will produce the green metallic sheen, and other gram-negative bacteria produce colorless colonies. So, eosine methylene blue agar is used for differentiation of Enterobacteriaceae, and it contains sucrose and lactose. And if the organism ferments sucrose or lactose, the pH will be acidification. So it will be acidification of the medium below 5, and it will cause the formation of methylene blue eosinate complex, which has a metallic sheen, right? Sucrose and lactose negative colonies are colorless. That's why. Can you see here? These are not colored colonies, non-pigmented colonies, and these are pigmented colonies, right? And this is the Klebsiella mainly, and that has negative reaction. So McConkie that you've seen already. So it contains crystal violet and bile salts that inhibit gram-positive bacteria. And you can see these are colonies of lactose fermenters like Escherichia coli, and these are colonies of non-lactose fermenters like Salmonella and Shigella. So then we may use bismuth sulfite agar for isolation mainly of Salmonella typhi because it produces hydrogen sulfide, and the growth of some bacteria, E. coli, Shigella, is inhibited of this medium, but Salmonella grow very well and give black colonies with a metallic sheen. And this we discussed already. Then quite often, for presumptive identification, we may use so-called triple sugar iron agar. That's very interesting medium that has 3 sugars. 0.1% of glucose, 1% of sucrose, and 1% of lactose out of the sugars, right? Then it also contains ferrous sulfate as an indicator for whether the hydrogen sulfide is produced or not, and also pH indicator phenol red. So this agar is used for initial differentiation of enteric bacteria. Salmonella and Shigella should be differentiated from mainly coliform bacteria, maybe Escherichia coli. So you can see that this is the intact TSI agar. It has the slightly pinkish color. Then how do we perform inoculation? We perform stabbing inoculation into the bud. This portion of the column is called bud, and this portion is called slant. So first we take a loopful of bacteria and perform stabbing inoculation into the bud, and then we inoculate the rest that we've got on the loopful here on this land. So we have anaerobic and aerobic conditions here. So anaerobic in the bud and aerobic in the land. And the relative use of each sugar and hydrogen sulfide production can be detected. If there's no changes occur in the bud and in the slant, then it means that sugars are not fermented. And these are called non-fermented Enterobacteria. So these are Pseudomonas, Acinetobacter. They cannot ferment sugars. That's why the appearance of TCI agar will won't change after the process of incubation. Then we see TSI agar after inoculation, being inoculated with Escherichia and some others, Klebsiella and Enterobacter. So you can see here the bud is yellow. It means the acid is produced out of lactose fermentation. And you can see here the medium just a bit jump up in the tube. It means because the fermentation happens with acid and gas formation. Not hydrogen sulfide, but gas, carbon dioxide. Then this land is also yellow. It means that the lactose in aerobic conditions it is also fermented. But then no clouds of no black clouds. It means the hydrogen sulfide is not produced. Here you can see the bud is the bud is yellow. Glucose is fermented without gas bubbles. It means acid, but no gas is produced. And lactose is not fermented because this slant is still red. So this fits with, and no hydrogen sulfide is produced. That fit very much with Shigella or Shigella in Latin. Then take a look at this slant. So that's typical for most Salmonella. So the bud is yellow. It means glucose is fermented. We don't see here with acetogas, no. With gas, we don't see really the gas bubbles. Then, slant is still red. It means lactose is not fermented. But then, here you can see the ability of these bacteria to produce hydrogen sulfide by decomposition of over the histidine and some other amino acids that contain sulfur. Okay, so this is most typical for salmonella. And some salmonella, like Salmonella typhimurium, may produce so much hydrogen sulfide that the the whole slant will be seen or will appear very dramatic black because overproduction of hydrogen sulfide. And see here, initially it was like this before inoculation, and then different types of Enterobacteriaceae may produce different picture due to the these components fermentation. So that is for initial presumptive identification of Enterobacteriaceae. But then we state that apart from biochemical tests, we need to identify the antigenic structure. The first one, that is the most important antigen, is oosomatic antigen that is associated with endotoxin or lipopolysaccharide. Why it is so important to identify? Because around 150 different oosomatic antigens are found on these Enterobacteriaceae. And that is the point by which we may distinguish them. So then this antigen is resistant to heat. We call it heat stable and alcohols. And even if we boil this bacteria, oosomatic antigen still will be detectable by some very simple serological tests called agglutination test, for example. So, the oosomatic antigen is the most external part of lipopolysaccharide, and it is made up of repeating subunits of uh, oosomatic antigens, and they can be detected by agglutination. Why it is important for medical doctors? Of course, you may never perform this agglutination yourself, but then it is important for doctors because certain types of diseases may be associated with a specific oosomatic antigens. For example, if we are talking about E. coli, oosomatic antigen, for example, 22 causes enteropathogenic disease, and oosomatic antigen 100, the bacteria that keep oosomatic antigen of 157, this is a number. So then it may cause very severe, most severe Escherichia coli infection that can be life-threatening. So that is a difference. So that's why we need to identify oosomatic antigens. The thing is that single bacteria may carry several oosomatic antigens. And one more thing to understand is that the members of Enterobacteriaceae may share same antigens within the genera. For example, E. coli may share all antigens with Klebsiella, Providencia, Shigella, Salmonella, and the others. Again, see, this is the lipid A, this is the core polysaccharide, and this is all somatic antigen made up of repeated around 40 repeated carbohydrate subunits, the side chains of the polysaccharide. But then, if you take a look at gram-negative Enterobacteriaceae, so here somewhere we have all somatic antigen, right? Right here. But then some of bacteria may produce capsule, as we said. And sometimes it may be microcapsule, very thin one. And the example is given for Salmonella typhi that has so-called VI antigen. It is a very thin layer, but this layer prevents phagocytosis of these bacteria, so that's why it is very important. Or sometimes, like in Klebsiella, it may have the thicker capsule like this, right? So then it also may prevent phagocytosis, and this is antigen that is external to compare with oosomatic antigen. And sometimes the capsule may cover oosomatic antigen, so we won't see it, and we cannot identify it in by agglutination. So then, to find the oosomatic antigen, we have to destroy the capsule first, and then the oosomatic antigen will be visible in the reaction. Then, bacteria also take a look. These are flagella, right? And it is made up of flagellin. And of course, flagellin, that is a protein, it is also immunogenic. So we call it H flagella antigen. And usually we have more than 50 different H antigens in Enterobacteriaceae. And they are heat labile and alcohol labile. We may remove this antigen by alcohol or boiling, right? And within a single or serotype of bacteria, H antigens may present in several forms. So that's because I don't uh, pay very much attention in the indication of how do we indicate. But then the thing is that the flagella antigens may be expressed or not expressed, and also because the information about the flagellum production may be written in different genes. Uh, but not in one gene. So the different genes may be expressed or not expressed. Then the flagellum antigen may vary. So that's why that is very important in what really form will we see the antigen. And that's also performed, identification of H antigens is also performed by agglutination test with anti-H antibodies. So, virulence factors that can be found of Enterobacteriaceae. Again, you can see Enterobacteria, my dears. I would like to tell you that generally all somatic antigens are not really virulence factors because the antigen itself, it does nothing for pathogenicity, the oosomatic antigen. But if bacteria are changing, keep changing the oosomatic antigens or H antigens, then it is important for the virulence because the antibodies produced against one type of O or K antigen will not recognize another O or K antigen. And this antigenic variability, then it can be considered as a virulence factor. But I would like to repeat, not the O antigen is a virulence factor. Now let's take a look at the virulence factors. What are the virulence factors? These are structures, proteins or other parts of bacteria or some proteins that are secreted by these bacteria that may cause harm to the body and will be responsible for pathological process in the body and tissue damage. So now let's take a look at the virulence factors. Endotoxin, my dear, again, lipopolysaccharide and exactly lipid A in this structure. That is a very important endotoxin that is found in all gram-negative bacteria. But it is tightly fixed with the cell wall. And usually lipid A is not directly exposed to the environment, right? Only when bacteria is autolyzed or destroyed by our immune mechanism, only then the lipid A can be exposed to the body fluids and body cells, right? Then it will express its toxicity. So, but then Enterobacteriaceae also may produce so-called exotoxins. So these are various proteins that will be released into the medium like this. See? These are protein molecules released outside bacteria, produced by bacteria and secreted to outside. So because they are secreted to outside, they are called exotoxins and they have particular targets on the cells, usually human cells. They bind with these cells and may act on different, according to the different mechanism on those cells. Then the capsule. It is recognized as a virulence factor because it prevents phagocytosis and interfere with the binding of antibodies to bacteria. For example, some capsules can be poorly immunogenic and won't cause even antibody production. Phase variation that we discuss. K and H antigens may be alternatively expressed or not expressed. So they interfere with the antibody-mediated bacterial cell killing by macrophages and others. Then almost all Enterobacteriaceae are able to perform sequestration of growth factors, especially iron. And usually Enterobacteriaceae may produce different systems. These are enzymatic systems to get iron from human cells because we don't have free iron in the body, right? So then change the valency of the iron in the hemoglobin or myoglobin and then transfer it into the bacterial cell. These are bacterial competitive iron chelating compounds. These are siderophores or aerobacteria. So then you know that bacteria also may produce antibiotic resistance. So that's why this antimicrobial resistance can be encoded by the R plasmids. So they resist drugs. So now let's take a look at E. coli as a very important member of Enterobacteriaceae family. So what kind of infections it may cause? Urinary tract infections. This is a type of E. coli that may cause non-complicated urinary tracts, especially in young women. And the symptoms are urinary frequency, dysuria, hematuria, pyuria, and flank pain, right? So they may result in sepsis, but most commonly they are non-complicated. What special virulence factors are attributed to them? This is P. pilus, very special one that binds to the P group substance. And those E. coli that have this factor, they are able to produce pyelonephritis because they move upward from the site of the infection and cause this pyelonephritis. Then they also have ephemeral adhesins, the molecules that allow them to tightly bind with the epithelial cells. And also they usually can cause hemolysis. So this is the pathogenesis, my dears. Now I will give some those pictures that explain the pathogenesis of Escherichia infections. You may see here that this type of bacteria may adhere to the uroplakin on the surface of the uroepithelium through their pili. See, this is a pili that's safe for adhesion. And then it causes actin-myosin interactions and the cell produce pseudopodia and engulf the or internalize these bacteria by receptor-mediated endocytosis. While it internalizes it in superficial facet cells, and then they start to multiply inside the cells, right? And they are the so-called intracellular bacterial communities are produced, right? Intracellular bacteria communities, right? So as soon as they multiply, they start producing hemolysis, and it leads to the host cell apoptosis and exfoliation of the superficial facet cells. Okay, so that is the idea. And then, of course, when bacteria are released, so they get better access to the transitional cells and may infect the deeper layers of the uh, underlying the superficial facet cells. Okay, so then this is the infection with uropathogenic Escherichia. And here you can see the result in pyelonephritis, how kidney appears as a result of the moving upward of the Escherichia to the kidney. So then other group of diseases except for urinary tract associated with the Escherichia are sepsis. This is when E. coli may cause, will be not killed by phagocytes, may enter the bloodstream, and usually it may happen when the immune system is not working adequate. So in newborns, for example, if they don't produce very important early IgM antibodies, you might know that IgM is the first antibodies produced, right? And as a result of the sepsis, when E. coli may circulate in the blood, it can be inside phagocytes that may keep them inside and will be incapable to digest them. They can be taken to the meninges, so to cause meningitis. And the newborns are highly susceptible to E. coli meningitis. So the major virulence factor of this bacteria is the capsule antigen. It is interesting that the Escherichia coli with this K1 capsule antigen are very similar and related in capsule antigen with that of the other gram-negative bacteria, Neisseria meningitidis, that may cause very severe meningococcal meningitis. Their K antigens cross-react. So it means this is something that is very common for them. So, and these Escherichia coli are called Neisseria meningitidis E. coli. Uh, they cause very similar disease, and we have the very similar K1 antigen. So how it happens? You can see here the uh, uh, Escherichia coli that has very heavy capsule that may inhibit the complement activation, that can inhibit phagocytosis, and the capsule may inhibit also dendritic cells maturation. All these three prevent the development of the proper innate immune response against these bacteria. That is the problem with these bacteria. They survive in macrophages and prevent macrophages to produce pro-inflammatory signals like interleukin-1 and tumor necrosis factor that help body to cause inflammatory response against bacteria. And what bacteria do? They bind with the CD48 molecule on the surface of blood-borne barrier, then and activating the actin rearrangement, it may act as the triggering mechanism for invasion of the cell with this bacteria. And due to the lysosomal fusion, then bacteria will be released outside the blood-brain barrier. So then they multiply and cause um, influx of the neutrophils on macrophages into cerebrospinal fluid. So the the pressure of the cerebrospinal fluid will increase. That is really cause symptoms of meningitis, very severe pain and the rigidity of the neck muscles and so on. Okay, apart from urinary tract and uh, uh, meningitis, we may have group of Escherichia that cause diarrhea-genic diseases or diarrheal diseases. And we all together divide them into 5 groups. And you have to know them and you have to distinguish them. Enteropathogenic E. coli, enterotoxigenic E. coli, enteroaggregative E. coli, enteroinvasive, and enterohemorrhagic. First 3 cause disease in the upper uh, digestive tract, right? And the second 2, the next 2, last 2, cause the diseases in the colon. Now, a few words about this. You can see this table, by the way, in the old lecture book for special microbiology. I would not read it in very details and describe it in very details, but I would like you to try to differentiate them by the disease and by the virulence factors that are produced. So the first one is enteropathogenic. That is the long-term infection of the enterocytes that cause destruction of microvilli and entering the back into mucosal cells. And usually we have the diarrhea that results from loss of absorptive properties of the infected cells. And usually main virulence factor are factors like adhesins, intermediate adhesins, and bundle forming proteins, also adhesins. And you can see the simplified picture of the pathogenesis on enteropathogenic Escherichia. So they bind with membrane proteins that are called intermins and then translocated into the translocation of the signal. Oh, here is it. So causes the signal transduction from outside the cell to inside the cell through several signaling mechanisms. What happens inside the cell as a result? Formation, the changing of the expression of the genes inside the nucleus of the cell, cause formation of so-called pedestals. See? Pedestal structures, when several microvilli fuse together. And usually a pet survive on the surface of this pedestal. And of course, this triggering affect the disruption of the microtubule network in the cell and stress fibers assembly, and of course it interferes with the normal function of the cells. And you can see here this formation of pedestals in the microphotograph where microvilli are destroyed already and the pedestals are formed, right? And this is normal with microvilli of these cells. Then enterotoxigenic. Please pay attention. Toxigenic, it means they produce exotoxin. And this exotoxin works as enterotoxin. So it means it causes diarrhea, and also very strong colonization factors allow these bacteria to stay attached to the to the uh, epithelium of the upper of the small intestinal tract, right? And the toxin produced by these bacteria cause hypersecretion of water and ions and inhibit the sodium reabsorption in the epithelial cells. How does it work? You can see that the Escherichia themselves toxigenic. They attach to the microvilli of the epithelial cells, and then of the upper intestinal tract. Then they start producing soluble factor, that is heat labile toxin, that consists of A subunit, 2A subunits, and 5B subunits. By B subunit, this toxin binds with the microvilli and will be uptaken inside the cell, and then from the vesicles the B subunit let A subunit to be released to outside, and the A subunit activate adenylyl cyclase, and it comes to the accumulation of the CAMP protein kinase there, and then it results in the release of water and some electrolytes there, sodium and chlorine ions, and then prevent the resorption of the sodium. So then we have influx of water, of liquid from the cell to the lumen of the gut. That is the result of the disease, loss of water. So then enteroaggregative. The major virulence factors are the ability of the bacteria to aggregate on the surface of the small intestinal tract epithelium, and there's a lot of adhesive molecules are there, including adhesive villi as structures. And then they also start producing enterotoxins that cause diarrhea, influx of water, and cytotoxins that may lead to the cell death. And the result will be the mucosa damage and secretion of mucus. So how does it work? Do you see here? So first we have adhesion of organisms and aggregation of organisms on the surface of the microvilli and the cells, right? And it prevents the absorption of fluid by epithelial cells of intestinal mucosa. And the result is a mucosa damage, secretion of hypersecretion of mucus, and sometimes due to direct toxicity of the toxin, cytotoxin that can be produced, that may get access to the blood vessels and cause bacteremia. That will be more severe. That were the E. coli that cause uh, infections in the in the upper intestinal tract, in the small intestinal tract, sorry. And now we take a look at two that perform infections in the lower or large intestinal tract. And these are enteroinvasive E. coli, and they cause disease similar to that caused by Shigella, with fever, cramping, watery diarrhea, followed by scant bloody stool with leukocytes release. And the virulence factors are mainly the invasion factors that allow enter-invasive Escherichia to invade epithelial cells. And this invasion usually causes destruction of the colonic epithelial cells. So what is the pathogenesis that's shown in the picture? They are usually intracellular pathogens that penetrate into the intestinal epithelium through M cells and goblet cells, right? And M cells. See, these are M cells. We have transcytosis through M cells. And then they're released and to submucosal layer, and they escape submucosal macrophages by induction of macrophage cell death. So while they die, the bacteria will be released, and followed by basolateral invasion of colonocytes. They let spread to the neighboring cells, and they really damage the neighboring colonocytes, and then the patches of colonocytes may be slotted over the surface of the basal membrane. So, and the last type is enterohemorrhagic E. coli that cause more severe disease, that are toxigenic also because they may produce toxins that cause disruption of the protein synthesis, Shiga-like toxin and verotoxin. It is cytotoxin will be called verotoxin because the toxicity of it had been shown on the so-called vero cells. These are the cell culture. So used to demonstrate the toxicity of some products. So this product was highly toxic for the cell, so that's why it was then been called verotoxin. And also Shiga-like toxin, common with Shigella dysentery toxin. But the disease doesn't like Shigella. So then, what happens? What is the pathogenesis of enterohemorrhagic Escherichia coli? So when they bind with the surface of the cells, of the enterocytes, uh, they alter the surface of the enterocytes, and they activate the coagulation cascade. They may go through the cells and released into the rich blood vessels, and there they may activate the coagulation cascade, which leads to the formation of the microthrombi and causes distal ischemic necrosis, platelet consumption, and red cells fragmentation. So the hallmarks of the hemolytic uremic syndrome. And this is a very severe state that may occur as a result of Shiga toxin activity over the enterohemorrhagic Escherichia coli. So, and this type of Escherichia even caused very severe outbreak in 2011 in Europe, in several countries like France, Spain, United Kingdom, Germany, and in this outbreak, the agent found was the recombinant of two pathogenic E. coli, enteroaggregative and enterohemorrhagic. And the strain has 93% homology with enterohemorrhagic Escherichia, but then they were they obtained the Shiga-like toxin from enterohemorrhagic Escherichia coli by the transfer of the gene that code for this toxin. And the surface antigens over the enterohemorrhagic, enteroaggregative Escherichia had been changed quite seriously. That's why they didn't recognize it. And out in this epidemic, 850 patients were uh, really under very severe conditions with hemolytic uremic syndrome, and altogether 3,500 with enterohemorrhagic gastroenteritis, and 50 patients died of the hemolytic uremic syndrome, and it occurs in 12 countries, and altogether 125 cases were documented, and a lot of them had been missed. So this is because still gram-negative bacteria exchange their genetic material very commonly with the help of mobile genetic elements like plasmids or extrachromosomal plasmids, pathogenicity islands, bacteriophages. So they exchange with the genetic material and undergo very intensive biological evolution. So from time to time, the recombinant strains appear that is not easily recognized as the cause of the diseases. That's what happened with Escherichia coli. So then we have discussed what tests we use for uh, identification of these bacteria. I won't repeat them again, right? So then you may see it when you read the uh, presentation. Take a look at the presentation again. But please don't forget that except for biochemical tests that are shown here, we need to perform serological identification. And since 2011, when we had this outbreak, okay, I would like to add that all uh, members of this diarrhea-genic Escherichia, if they are isolated from the patient, they should undergo the study of the genome by polymerase chain reaction to find whether some genes that code for the toxins are present there or not. So this is the recommendation for identification of E. coli, to use polymerase chain reaction to demonstrate the presence of the absence of the genes of pathogenicity. So now a few words about Shigella. It is a genus of bacteria that cause quite severe gastroenteritis, and Shigella dysentery causes bacillary dysentery. They are non-motile, non-sporin, and non-capsulated, and they are indistinguishable under light microscope from other Enterobacteriaceae. And they are similar to most other Enterobacteria. They cannot ferment lactose. They are very weak fermenters. So, but then Shigella dysentery itself may produce the toxin that is called Shiga toxin, that may cause the cell death. This is cytotoxin. But then apart from exotoxin, all Shigella have very strong invasive properties that are related to the presence of particular plasmid that code for outer membrane proteins responsible for cellular penetration. So when we classify Shigella, we distinguish four species of Shigella. Shigella dysentery with 10 serotypes regarding all somatic antigen. Shigella flexneri, 6 serotypes. Shigella bowdii, 15 serotypes. And Shigella zoni, serologically homogeneous. It means the antigenic structure is stable and we don't distinguish any serotypes there. The disease caused by all Shigella except for Shigella dysentery is shigellosis. This is a quite serious infection of the bowel, marked by diarrhea, stomach pain, fever, sometimes if especially we have bacillary dysentery, and that is carried by hand-to-mouth contact with the feces of infected individuals. Uh, Go ahead with the pathogenesis. See Shigella bacterium. So they may travel, they may make M cells to engulf them, and through transcytosis, they should be transmitted uh, under the layer of the epithelial cells in the lane tissues. And usually macrophages engulf them, but usually macrophages cannot kill them. And they may survive in macrophages and cause apoptosis of macrophages and initiation of inflammation. Then Shigella bacteria also can activate NF-kappa B factor caused by intracellular location of shigellosis and the release of some interleukins that may inhibit the neutrophils' activity. Then entry of Shigella mediated by type 3 secretory system and other effector proteins cause the skeletal rearrangement, and then it may cause the infection of traveling from cell to cell. They may cause the infection of the neighboring cells. Then we may have the damage of the epithelial cells of the colon, because that happens in the large intestinal tract, and sloughing of this layer. So then, the next one is Salmonella. And this is a genus of bacteria that may lead to several health conditions. The first one is typhoid fever caused by Salmonella typhi. Then enteric fever caused by so-called typhoid bacilli. This is Salmonella paratyphi A and B. And then the rest are Salmonella or non-typhoid Salmonella. Quite often we may see them under the abbreviation NTS, non-typhoid Salmonella. But still they may cause gastroenteritis as a result of visiting, for example, shawarma cafe, and in rare cases, septicemia. And all of them may cause carrier state. Usually they are motile, facultative, anaerobic, gram-negative bacilli, and they're able to grow in the wide range of simple media, but also, of course, we use quite often a selective media, especially if we isolate them from the feces. Then, if you take a look at Salmonella, this is Salmonella typhi in triple sugar iron agar, and this is Salmonella typhimurium. So the production of the hydrogen sulfide is very typical to some Salmonella. Usually they are non-lactose fermenters, and they are good fermenters. They can produce acid and gas out of glucose, and they are identified by biochemical characteristics and antigenic structure. So classification of Salmonella is very, very complicated. Altogether, we have up to 2,000 unique serotypes that can be distinguished by antigenic structure and by biochemical activity. So, but then now these 2,000 unique serotypes belong to just two genera. two species. Salmonella bongori, but then this salmonella cannot cause diseases in humans. And Salmonella enterica, that is a collection of all salmonella that may cause disease in humans. They are considered as same species, Salmonella enterica. But then, according to the biochemical properties and antigenic structure, Salmonella enterica is divided into 7 subgroups with 2,000 unique serobars. So you can compare the uh, Salmonella typhimurium and Salmonella typhi and Paratyphi A, Paratyphi B. So then you can see that the structure is very much same, right? Peptidoglycan in the periplasmic space, then inner membrane and outer membrane. Then Salmonella typhi produce the unique layer that is called V capsule. Don't forget, please, see? And it is called V antigen. That is shown here, see? That is not shown in Salmonella typhimurium. Of course, both like other gram-negative bacteria have lipopolysaccharide. Then they use also Fimbria pili for adhesion. So then, of course, in both we may see the Salmonella pathogenicity islands, and they are all peritricious like this, like they have flagella and flagellae. And they also may have pili of type 3 secretory system to inject the proteins from the cell into the environment or into the host cell. So a few words about Salmonella. We may have typhoidal and non-typhoidal Salmonella. The sanitary water and food supply is very important in prevention of Salmonella and eradication of organisms in the carriers. Salmonella may live in many animals, but only non-typhoidal bacilli. Typhoidal live in human body only. So then, it can be transmitted, typhoidal can be transmitted mainly through contaminated water, and if this water contaminate food or whatever, vegetables or fruits or whatever, and if the food contaminated during the cooking by cooker is not cooked properly. So, but then those that live in animals may contaminate meat or poultry meat. So, and they usually live in animals. These are non-typhoidal bacilli. So then they can be transmitted to a new host by directly caring by the uh, meat. So if it is undercooked, that can be a source of these non-typhoidal bacilli, or poultry eggs that we use undercooked or use them not cooked in the creams, in the production of creams or so. And also they can be carried by those who prepare food. So if we have the oral infection, so we have salmonella that enter through the M cells that get them and move through the epithelial cells. And through M cells, then they may enter the macrophages and dendritic cells. But then, quite often, they may reside in the rest in macrophages. and they may also activate macrophages to produce the activated interleukins to accumulate polymorphonucleated cells. But then through traveling inside mainly macrophages, but mainly dendritic cells, but also macrophages, they may enter the mesenteric lymph nodes, and from the mesenteric lymph nodes, bacteria may travel and may get into the bloodstream, where it may be carried by many macrophages and they're released freely. Then it may cause systemic bacterial spread and mixing of bacterial population between organs, mainly lymphoid organs, liver, spleen, and bone marrow, by the way. From there, they may get to Kupffer cells and polymorphonucleated cells, and inflammatory macrophages may occur. And they're also through the red pulp and marginal zones of with macrophages, they may cause the very strong inflammatory response. So usually salmonella infections usually are accompanied with very high intoxication and very high temperature. So, but then if you compare the pathogenesis of salmonella infection that cause gastroenteritis and typhoid fever, there's a big difference because typhoid fever really is a long-term infection, and the incubation period is around 14 days. What does it mean? That bacteria travel 14 days in the body before the symptoms appear. And here you can see that what happens with Salmonella typhi getting into the body, getting into the macrophages, then getting into the bloodstream, and this is primary bacteremia, then spreading in the bone marrow, gallbladder, spleen, and liver, and where they multiply again. See, my dears, by now we don't see any symptoms because there were not so many of them initially, and they don't multiply directly in the gut. They travel inside macrophages to the target organs, and then they start multiplying. Finally, they are released back into the bloodstream. So we have secondary bacteremia. My dears, then we have symptoms appearance. Why? Because when they get back to the bloodstream, the antibodies are already produced. They cover Salmonella typhibacilli, and with the help of macrophages that can see the bacteria are marked with antibody, that is called opsonization. They start killing these bacteria. The endotoxin is released. So then it is the generation of the temperature happens, fever and very severe state that is called endotoxemia, when the endotoxin is spread through the bloodstream. That is the beginning of the disease. Then we have widespread dissemination, and then they also may reinfect through gallbladder. They reinfect the gut, then they infect the Peyer's patches, and again the in the Peyer's patches, the very strong inflammatory response may occur that may lead to the damage to the Peyer's patches and perforation of the colon, and that is a very severe complication of the possible infection with the typhoid bacilli. That's a bit of immunology. I will skip this because you didn't study immunology yet, and we used to teach also students that studied immunology. And there's a bit of history, my dears. You may take a look if you type typhoid Mary in the and ask Dr. Google who was that. So that was Mary Mallon. There's ages that also known as typhoid Mary, who was the first person identified as a healthy carrier that carries Salmonella bacilli in the gallbladder. So, and she was presumed to have infected some 53 people because she had been working first as a babysitter and then as the cook in the restaurant, and that's how she had been shown as a cooker in the restaurant, so throwing scalps on the pan, right? And she was forcibly she uh, mm, the doctor suggested to remove the gallbladder where she carried bacilli, but she rejected. So, and then she was forcibly quarantined twice by public health authorities and died after 3 decades in quarantine. So, that's why the carriers are very important in spreading of this disease. So, there are several types of anti-typhoid vaccine that can be used in prevention of this. So the whole kill vaccine, the very old one called TAB, vaccine against Salmonella typhi, paratyphi A and paratyphi B. Then the newer one is the oral live vaccine. This is stable mutant of Salmonella typhi, and the vaccine is called Typhoral because it is given orally to cause the mucus immunity on the mucous membrane. And also the chemical injectable vaccine made up of V capsule antigen of Salmonella typhi also can be used for immunoprophylaxis of this disease. Okay, guys, I think we may finish with this, and I hope it will help you. This lecture will help you in the uh, special microbiology. This is last lecture for first division in this semester. My dears, goodbye and good luck on your examination.